Welcome to the short audiovisual abstract of our article. Our work is titled Cardiovascular Brain Impulses in Alzheimer's Disease, which implicitly includes two main statements. First, with refining our previously published method, we can now measure the 3D propagation of individual cardiovascular brain impulses within the living human brain. Second, we show how these pulses are changed in Alzheimer's disease and discuss what implication this has on brain clearance. The lymphatic system is a network of vessels that clears waste from the central nervous system. Among other forces, lymphatic transport uses energy from the pulsing of arteries. The waste products such as metabolites and proteins are transferred out of the brain via cerebrospinal fluid (CSF). Lymphatic brain clearance impairment is linked to amyloid beta accumulation, which is one of the characteristic features of Alzheimer's disease. It has been shown in the brain that flow of CSF is driven by arterial pulsations. In this video of an animal model, we can see how CSF flows in synchronization with arriving cardiovascular pulse wavefronts. This is the effect of cardiovascular impulses on the microscopical scale. Mechanical energy is also needed to move waste particles in the brain. Continuous mechanical impacts are provided by the physiological pulsations, namely cardiovascular impulses and pressure changes induced by respiration. As arterial pulsations have been shown to drive CSF, we developed a method to measure propagation of cardiovascular impulses within the brain. To measure physiological signals in the living brain, we need a bold functional MRI sequence so fast that its sampling frequency can keep up with our heart rate, technically speaking the double of that. At the cost of reconstruction time, the MREC sequence samples the full brain at a stunning 10 Hz. This means we can measure without aliasing up to 5 Hz, which is conveniently above the cardiac frequency of humans. With just a few more steps and our multi-resolution optical flow method, we could calculate the propagation speed of the cardiovascular brain impulses voxel-wise in 3D. We have 5-minute long resting state full brain recordings at 10 Hz with a velocity vector field for each frame. We selected data to build up a model for a typical cardiac cycle and compared the following properties between Alzheimer's disease and control groups: the arrival time of the impulses, the magnitude of pulse propagation speed, and the directional comparison of velocity vectors. We found the most intriguing results by analyzing the direction of the impulse propagation velocity vectors. We were interested in the direction u for every voxel onto which projecting the velocities, the group difference between Alzheimer's and controls is maximal. In other words, it means that looking along that calculated u direction, we see the biggest change in Alzheimer's disease. We got a nearly perfect mapping of arterial territories within the brain at the time of cardiac impulse arrival. The U directions follow brain vasculature, so cardiovascular impulse propagation is changed in Alzheimer's disease according to local vascular structures. In our opinion, some pieces of the puzzle are connected here. Cardiovascular pulsation is indeed different in Alzheimer's disease on the macroscopic level. Arterial pulsation is linked to CSF flow on the microscopic level, which in turn plays a role in brain clearance. With the mapping of the directions for every cardiac phase, we can group the velocity changes into the following cases: areas where impulse propagation in Alzheimer's disease is faster, red, slower, blue, and the green areas where impulse propagation is reversed in Alzheimer's disease. In these areas, looking into the direction u of maximal difference between groups, the sign of propagation speed is inverted. In practice, this means that in the neighboring areas, the phase of impulse arrival is mismatched or shifted, so the pulse wavefront propagates from neighbor B to A instead of A to B. The pulses get reflected from narrowed arteries, which might cause shear and physical stress on the blood-brain barrier between the lymphatic paravascular space and blood vessels. The areas most affected by this are in the border or mixed zones of arterial territories and include both hippocampi as well. Let me wrap up with the conclusions. There is perturbation of cardiovascular brain impulse propagation along the cerebral arterial territories of Alzheimer's disease patients. Proximal arteries seem to be dilated, while peripheral vascular territories showed increased impulse speed. We observed directional reversals of the impulse propagation in regions typically showing elevated amyloid beta accumulation and blood-brain barrier pathology in Alzheimer's disease. The abnormal impulse propagation may show impaired vascular flow that predisposes to neurodegeneration and cognitive decline in Alzheimer's disease. Thanks for watching and check out our article for the details.